Yeah, I can move the RStudio around a bit. Um, what I have done in the past is I have I've made a scene that focuses in on one part of the RStudio or whatever IDEA I'm using window, like the code or the terminal or the graph window. But then I always so it turns out that it's a little irritating to do that. All right, I'm going to go ahead and start. I think I did put the link in here. I made this a little bit bigger because I wanted to say that I've added a couple of topics for the next few weeks. And I wanted to just say a word about it. Recently, I was working with Joe and Eric and Megan. Actually, it wasn't that recently. Now that I think about it, it was about a year ago. We were working with somebody. I won't say who they are or where they were located, but they were a fully grown adult scientist who was talking about farm data. And there's always this issue with farm data that farmers don't want to share their data because it's commercially sensitive. It might have IP. It might have um, things in it that they don't want to share for various reasons. And there's a modern solution to that, and the modern solution is called synthetic data, and it's a simulation technique that statistically removes 100% of the information from a data set. It works best, of course, if you have a large, robust data set with many, many data points. An example of this might be if you were studying dairies and you had lots and lots of dairy data from lots of farms in a lot of different countries, and they all did things their own way, but you had a cohesive data set. But you needed to retain the intellectual property um, for a particular farm to conceal it. And this method of synthetic data removes the individual information and recreates the statistical information. So you can have infinite simulated data sets that can be analyzed just like a real data set while removing the individual farm, but still making generalities about country, region, farming method, and so forth. It is actually not a huge um, it's not a huge method in agriculture, but I think there is great scope for applying it to agriculture problems because of this issue with farmers sharing the data, and something that I talk about in here sometimes that I call the environment by management problem. The environment changes across management changes every year, even in a single farm. But for example, in human medical health, synthetic data has absolutely revolutionized the data sets and statistical advances, and therefore the scientific advances that we've made based on large patient data sets. Cancer research is one that I'm thinking a lot about these days. And I even reviewed several synthetic data papers for diagnosing cancer within the last year. And anyway, I want to talk about synthetic data and just introduce the idea um, for my own benefit and that of others to get you thinking about the possibilities for synthetic data, not just using it, but possibly even contributing to synthetic data sets. But I was astonished when I was explaining this to my colleague in this research project that they just completely missed the point and pronounced this. This is a verbatim quote, as far as I recall. Farmers don't care about the fake data. And so I thought, yeah, there's probably a talk in there where I can explain why they very much should and why it's not fake data. And then the week after that, I thought I'd go to a topic that is evergreen in here, especially for first year students, but actually it is very relevant and important for all ages of scientists. And that's what would the sample be for your experiment? What should your sample size be? And this has to do with statistical power, experimental design, and effect size. So this will be like an introductory session with some code where I'll introduce that. The synthetic data will be conceptual, but I hope to actually synthesize some data as an example in that. What are we doing today? Well, today I decided to talk about, Megan and I have been working on some biodiversity data. There's several biodiversity initiatives around campus. We've been working on a few of them that are part of, well, I guess they're all loosely related to agri-environmental concerns around campus, but they have more to do with stewardship with the data sets we've been working with, rather than explicitly opting into for-pay schemes. And one of the things we've been struggling with is we've collected a lot of data, our colleagues, that is, have collected a lot of data around campus doing monitoring to baseline biodiversity. And we've been talking about how to create context. So I'm going to tell you about a source of data, and really, this is a huge iceberg underneath the ocean, and we're just going to scratch the very top of it today. And if people are interested, I would absolutely love to give more sessions digging in deeper into creating information with biodiversity data. But we're going to start today. I'm going to give a little bit of a talk to start with. You can follow along either the old-fashioned way on the PDF or on Canva if you want, or you can just watch me. And I've got a link later on I'll show you to the GitHub where you can get some R scripts, and I'm going to run through as many as we can get through. I've got a fairly ambitious set of things to go through, so slow me down if I'm going too fast, and we may not get through all of it today. And do let me know if you're interested in this topic, because I would love to do some more sessions on it. And not only that, Megan has been working very hard on some things, and maybe in the future we can showcase some of that and talk through some of the things that she's created to display and create information with our local biodiversity data. Okay, so without further ado, I'm just going to go to the Canvas slide. I already have it open. I'm just going to start it. So I thought I'd start really broad with this and pose the question, what is biodiversity? And I pose this question, I don't pose it lightly. The reason I pose this is that people, scientists that is, who are specialists in studying biodiversity in all aspects, not merely the act of measuring biodiversity by counting species, but people who study the source of biodiversity from an evolutionary perspective on the planet. People who study the sources of biodiversity relative to ecological processes and relative to, let's say, some of the alternative hypotheses include that there is more energy hitting the Earth in certain parts, and therefore there's a larger base to support the evolution through millennia of biodiversity. Disturbance. There's an intermediate disturbance hypothesis that disturbance breaks up ecological connections and allows succession. That's in ecological time, but in evolutionary time allows the proliferation of species. There are many such hypotheses. So people who take this stuff seriously spend their entire lives thinking about it. The reason I pose this question, what is biodiversity, is because they themselves argue about what is biodiversity. And I'm just going to go through a couple of the simplest ones. You probably heard of all of them, maybe not all of them. Maybe there's something new on this slide, but the simplest way would be to count the number of species that occur in a given place, a given time, a given habitat, or so forth. We often think of this, especially people who are not biodiversity specialists, and there's a jargon term for the number of species, and we refer to it as species richness. And along with species richness, there is a notion that it's not just the number of species. It may be something to do with the distribution of species, because in some places, species may be relatively evenly distributed in different species, and that may seem more diverse, may seem relatively diverse, as opposed to a different place that's dominated by one species numerically, but it has the same number of species. That would seem less diverse. There's functional diversity. This is one that underpins some of the agri-environmental schemes that we talk a lot about around Harper, where we're interested in specifically aspects of biodiversity that support habitat functions. And this would be things like trees and grass and soil structure that supports wetland, or ancient forests, or functional grasslands that support a lot of pollinators with a diverse flora. And yet, other people focus on genetic variation, literally the genetic variation that's contained within populations and within species. So if we think of a species that I've worked on in the past, the common dormouse, or the hazel dormouse as we call it in the UK, the hazel dormouse has a very wide range across Europe, and it's considered it's a highly protected species in Britain, and it has declined over about 100 years, and there's real good evidence to show that. And if you study the genetics, which I have, of different populations in Europe and in Britain, what you find is that as populations of all species break up and have become smaller as human activities have increased, literally the amount of genetic variation captured in a single population has decreased constantly over time. And if the population has a big range, it hasn't reduced down to the same genetic variation. So the greatest amount of genetic variation is not within a little population like the population of all dormice in Britain, relatively small population compared to the global population. But the biggest variation is between populations that are now separated. So it's questions like this that drive questions about genetic biodiversity. And then finally, there's a question of phylogenetic diversity. You know, and here's an example question that people ask. If we've got all these species out there, and you know, let's just say we're talking about reptiles and amphibians. We want to save as many species as possible. We want to save all the species, right? But we can't. We have limited resources. Habitats are degrading. We're having extinctions every day, every hour. So is there something else we can do to help us make the decision in a triage situation? And the phylogenetic concept, there's an acronym. I won't be able to conjure up what the acronym stands for. It's called the EDGE concept, EDGE, that has to do with phylogenetics. What it posits is that maybe we should focus on unique phylogenetic signals that took a long time to evolve and are distantly related to other species. And those are ones we should say first. An example of this, a famous example of it, would be the tuatara in New Zealand, or some other group that maybe only has one or two species in a family. We would save those first, because in another family, there may be many, many species. And if we lose one or two, evolutionarily speaking, information-wise speaking, we've lost relatively little. At least that's the concept. Now, when non-specialists talk about biodiversity, we're often just talking about occurrence data, what is called occurrence data. This is a place where someone has measured in some particular place or time an individual from some known or identified species or taxon. So example would be, you go outside and you see a wren, take a GPS coordinate, note the date and the time, This would be an example of a single occurrence data point. And these kinds of data sets are very, very common. They comprise probably the vast majority of biodiversity data that are recorded. And they are the predominant kind as well for agri-environmental schemes and other citizen science and other practical conservation-based schemes. I thought I would also tip the hat as to why we care. I probably don't have to do that. You've probably heard some of these reasons before. But the people who specialize in biodiversity have thought very deeply about these questions. So why care about biodiversity? Well, probably my favorite one is the biophilia hypothesis. I don't know if you've heard of this. This came out when I was approximately in grad school. I don't remember the year. I remember reading it then. It was co-written by the Nobel Prize winner Edward O. Wilson. E.O. Wilson got the Nobel Prize not for biodiversity or the biophilia hypothesis. Does anybody without Googling know what E.O. Wilson got his big prize for? Anybody drop that in the chat? Any entomologists possibly? No? He got it for, well, I'll give somebody another 10 seconds while I ramble on too, in case somebody does know about E.O. Wilson. Ants. He did get it about ants. Got it about chemical communication in ants. I'm really happy that someone knows that. He's actually an excellent human, other than his research on ants, though. He has written a lot of things over the years about many topics, and one of them was this hypothesis that humans evolved in the context of nature, and as an explanation for the physical and psychological ills that mankind seems to suffer from, possibly increasingly in recent times, that it's because of breaking up the connections between our environment that we evolved in for millions and millions of years, humans maybe 4.5 million years. And then in the last few hundred years, that environment has been completely altered in a radical way. And it's breaking up these correlations between our phenotype and our environment that might cause those ills. And this is referred to generally as the biophilia hypothesis. We also care about the ecosystem services, stuff like stuff we think about a lot here on Harper campus and in the agriculture world, like pollination, pest control, food stabilization. I see some people in the chat I know that could tell me a thing or two about all of these topics, but I consider them very important. Ethics, such as just the belief that all species deserve to have a natural course in their history, their natural history. This is probably the one that is the most controversial, strongly held by some, completely disregarded by others. Linked to ecosystem services is one that is the economic value of biodiversity. And this could be for different reasons. It could be for ecosystem services, but it could also be for tourism. I don't know if anyone here has been lucky enough to travel as a tourist or be from, I'd see some people that are from East Africa, where there's a massive tourism, biodiversity, wildlife tourism industry. It's the country I'm most familiar with is Tanzania. They're, for a while, even bigger than agriculture was tourism, and I'm not sure exactly where it is, but it will be close, I believe, for Tanzania and probably some other countries in East Africa and maybe in other places as well. But also, people believe there could be future crops that we have yet to discover contained within the black box that is the unmeasured aspect of biodiversity. And E.O. Wilson himself speculated that, and that many people have speculated about this, that there have been estimates that we only know about between 1% and 50% of extant species. And probably the truth is somewhere in there in the middle. maybe these days it might be 50% for some taxa like vertebrates, but for plants, bacteria, it's surely many more unknown species. And so it's the economic value for future crops and possibly pharmaceuticals that can be derived from yet to be discovered or yet to be exploited species. We could put it that way. And then finally, environmental concerns. If biodiversity degrades, many have measured effects that systems of soil and oceans and ecosystem functions, such as clean air produced by the Amazon rainforest, break down. And again, this is of course related to ecosystem services. So how do we measure biodiversity? Well, we mentioned a few of the things. I've put up a few papers that I knew of and had in my library that are citation classics. This one probably has more than 1,088 citations. One of my mentors in the past, I often quiz people about, well, what makes a good paper to you? How do you call out a good paper? And if you discuss this for a short period of time as a scientist, you might decide that, well, it could have something to do with where the research was done, how good the research is, who did the research and how good they are, how well known they are. But you will always come around to the problem of, well, how do you measure those things that are all subjective? And one objective way to measure it is how many other scientists have considered the work powerful enough, important enough to cite it themselves. So one of my past mentors said, if you have 10 citations for a paper you write after a handful of years, you've probably done a pretty good job of citing yourself. And if you have 20, you probably have given a talk at a conference and one or two people have cited you a number of times. If you have more than that, if you have 50, you're starting to make some impact. People are paying attention to your work that you haven't directly contacted. If you have 100, that's pretty astonishing. If you have 100 citations, 100 other papers have been written citing your own work. If you have 1,000 or more, this is very rare. So this is an important paper. Anyway, I've cited a few of these papers in this talk in case people are interested. But how do we measure biodiversity? I mentioned richness versus evenness. Both of these pictures have the same number of species. Oh, actually, no, they don't. This panel has 3 species, and this panel has 2 species represented. Richness versus evenness. If we were to compare these panels based on richness, we would say that sample A has higher biodiversity. But if we were to take a different measure, one related to this evenness measure, and say, if you pick any 2 organisms out of each of these samples, what's the probability that the organisms are the same species? And what you'd see is that sample B has a much higher probability that you get 2 different species. And that's a way of thinking about evenness. So in the sense of richness, if we're just counting species, this is more biodiverse. But if we're talking about evenness and the perception of biodiversity, the distribution, sample B is definitely better. And then I have something over here from this paper that shows the proportion of species that are known versus thought to exist. And the little circle on the inside of these pie slices, apologies for the pie chart to everyone and my ancestors. But the inner circle here is the proportion of known species in each of these taxa. And the biggest one by far is, as you can see, the insects. And another big one over here would be fungi, and another one would be bacteria, and so forth and so on. Chordates are relatively small, little sliver in terms of number of species. We also see the proportion on the outside of the circles that are thought yet to exist but undiscovered. And so for insects, it looks to me like I'm assuming this is an area rather than a linear distance. it looks to me like this is about 80 or 85% yet to be discovered for insects. Whereas if we look at plants, it looks like people think most of the plants have been discovered already. If we look at chordates, about 50-50. And I guess most of the ones that are undiscovered might be fish, non-terrestrial chordates. So just some patterns. This is kind of a busy graph, but again, it's a relatively important paper, and it talks about the efficacy of agri-environmental schemes. I'm not going to pour in this paper because there is such a huge literature for these agri-environmental schemes over the past 20 years across Europe and in the UK. This is a big deal because this is some real heavy hitters in the conservation world that are putting all of their strength and intellect into figuring out, are agri-environmental schemes beneficial to us? And Bill Sutherland and Lynn Dix are just giants in the field, and they're both based in the UK. What I want to communicate without spending too much time on these graphs are that, one, if we look at different countries across the EU, this is a bit of an old paper now, before our glorious liberation from the EU, I suppose I'd say it that way. It shows the countries at that time in the EU, and it shows the utilized agricultural area relative to the expenditure in converted from the local currency to millions of euros. What we see is that some countries, France, Germany, and the UK, have quite a large agriculture area, and they're spending a massive amount. And then there are a bunch of countries that are barely spending anything. I wonder, I'm looking at my Polish friends in the chat. I'm sure Poland has changed because of the rising fortunes of your country. I wonder where it sits. It's just a giant in agriculture and a real giant in economics and rising all the time. But the real important thing that they studied in this paper is this has to do this thing called HADC that is called effect size. This is a measure of difference. It's a statistically unbiased measure of difference if you're comparing usually two different things. And here what's being compared is the general effectiveness of agri-environmental schemes for two different time periods. And what they find is, now by HADC standards, these effect sizes are pretty decent. They're pretty decent. There's quite a big error bar around the second one. And a thing that I note here is that just for this last little bitty period, that there's been a massive amount of studies compared to the first long time that these schemes were in place. And one of the things they make a big point about in this paper is this panel over here, where they're talking about the impact it has on species diversity. And these comparisons are not the year, but it's comparing the effect size for areas that are measured in production, which is the vast majority of the studies across the entire study period, I think 84 to 2009, versus out of production. And what they argue is that this basically just shows that the massive impact that agriculture has on the reduction of biodiversity. I'm not going to spend more time talking about that. It's very complicated. And a thing I would say also is that there are data sets that argue that there's a big positive effect of these schemes, and there are other data sets that argue that there is no effect, or that there's even a negative effect of the schemes. So scientists believe that the jury is out as to whether these schemes actually work. All right. What are we talking about today? I've spent so much time talking that we are going to have to pick it up, and I'm almost through all of my slides. But I want to tell you about a wonderful resource. It can be used to create context, and actually it can be used for independent novel analyses if you're interested in biodiversity, if you're interested in that kind of thing. And the resource I'm talking about is called GBIF. If anybody's heard of GBIF before, would you put a 1 in the chat? And if you've never heard of GBIF, put a 0 in the chat. Let's just take a little bit of a survey. Now, a lot of people who have used this kind of stuff and worked on biodiversity problems before may have heard of GBIF before. It's kind of a big deal. I'm thinking of that, can't remember that guy's name. Yeah, I'm kind of a big deal meme, because it's like that. If you work on biodiversity, you probably know about GBIF. What it is it's a heavily funded international network of all sorts of different databases that are in a completely coherent standard format called the Darwin Core. One of the amazing things is it's free to access. I hope I'm able to demonstrate that in an elegant way to you in the time we have remaining. And it covers all types of taxa. I've got another paper here. There are many, many good papers in great journals that are highly cited about GBIF. I think I published my first paper using GBIF data about 10 years ago. I published a handful of papers using GBIF data. So I know a few things about it. And recently, when I was thinking about this and working on some things with Megan, I realized that it has changed over the years. It's gotten much better, for one. And another thing I realized is that the tools I used to use to access it have also changed. They probably have also changed for the better, but one finds it a little frustrating when the tools you're used to and remember change. The thing I want to say about this paper is that there are sources of bias built into GBIF that we need to take into consideration, because these are citizen science. They're high quality data, but they are citizen science data. And we have to make certain caveats and make certain adjustments to balance the bias that's inherent in the data set. So one of them, in this panel A, what this shows is the proportion, or the total number of observations of animals in different taxa. And you can see that the largest number of observations is birds. And if we go down, fish are up there. Plants of different kinds, trees, arthropods, bivalves, arachnids, and we get down to an ambiguous other category. I wish someone could tell me what globothalamia were, because that's just beyond the edge of my taxonomic awareness. So there's a massive bias. We know that birds don't dominate the number of species. How many birds are there in this world without Googling? Does anybody know approximately how many birds there are in this world? Yeah, there's about 10,000. Excellent. Here are some graphs. Now, I'm not going to spend much time in these graphs, but I do want to make one point that summarizes these other points over here. If you look at the records, B is the total number of records in GBIF, and we can see some hotspots like North America across the world. But if we weight those by the number of records standardized by the size of the country, you can't really see it because GB is so microscopic on this map, but we're punching above our weight. We have more records relative to our landmass area than others. And then this last one is the total number of records per capita. In general, what these second two graphs show is that in tropical areas, and especially in Asia and in Northern Africa, that the GBIF records are underrepresented, So that's just a pattern to show. But these sources of bias, the fact that some taxa are, why would birds feature so heavily? It's a hypothetical question. I'll go ahead and answer it for you. It's because they're so easy to see and people watch them a lot. They make noise, they flit around, they're colorful, and it's become a thing. And to watch birds relative to other kinds of species, Another thing that I'll show you in a second is the source of the data from different databases. Now, you can download different kinds of data from GBIF, and they do have a web interface. But you'll know if you've spent any time listening to me talk about documenting data collection that if you have a database, we need to document the capture of data programmatically so it can be exactly replicated. GBIF makes that relatively easy. I say that with a little tongue in cheek because it's a very complicated, powerful tool, but they do make it possible is the true way of saying it. They do have a web interface. This is sort of the amateur interface, but it is still useful, and it can be used to document real analysis if you're very careful to capture data through their interface, but it's much harder to replicate than if you create code in R or in Python. So this is an example of one of the databases called the occurrence database. And I'll just give you a little walk here. And one of the aspects of this is you can select an area. So here I've selected a bounding box across the United Kingdom. I've selected a subset of the records that include coordinates, and I haven't selected anything else. So this is all the records, all taxa, all years. And by the way, the years go back a really long way. You'd be surprised. If you want to check out GBIF, it's really fun to play with this and just give it a 30 minutes to explore it. But you'll be very surprised at how old some of the records are if you look. Now that we've made some selections, very few in this case, whoops, what we see over here is that we have a series of individual records. So someone on the 25th of January saw this individual species at these coordinates. And there are about, I think there are 80, or depending on how you divide the Darwin core data grab, the typical one has about 80 columns of data, including all sorts of taxonomic things and time and place and codes for location and other things. Now, all the records in the database, from just excluding ones without coordinates and just choosing the geographical basis of the United Kingdom, crudely as I have here, there are 215 million records, individual occurrence records. I think that's quite astonishing. And if we look at a slightly different view, where I've done a little bit more selection, been a little bit more careful, here I've picked birds, the aves, the technical taxonomic way of choosing it. I've picked the years 2000 to 2025. I have chosen the built-in, the baked-in shape, geographical shape that is Shropshire. This is, I think this is called a GADM code. I had a little trouble with these thinking about today. The thing I really want to show you about this is that there's one, since I've made these extra selections, there are vastly less results. So for this 26-year period inclusive, we've got 250,000 birds in the database. But the most interesting thing is where the data are coming from. And I think for a lot of records like plants and insects, iNaturalist, I hope some of you know about iNaturalist. If I do another survey, perhaps Claire will say you know all about it, and I hope you do. More power to you. I hope some other people also know about it. It's a fantastic app. It's a way to record species observations that you make. And this is research grade iNaturalist observations. And I think what that means is if you make an observation of a species, typically you take a picture of it. And if someone else corroborates your identification, who themselves is listed as a research grade participant, and I think that they become research grade participant if you have like 10 or 25 independently corroborated IDs that you make. So these are validated ones. I thought iNaturalist would have been the biggest one, but the vast majority, as you can see, is eBird. I hope you know about eBird, this vast scientific, citizen science, crazy data collection experiment. It's been going on for around 15 years, I think, at Cornell. It's out of Cornell University, but it's a global phenomenon now. This accounts for the vast majority of all the bird records. And the nice thing about eBird is I believe that almost all of the observations have a picture, and they're all at least as good as the iNaturalist research grade. I know they're better because I know that eBird I used to be friends with, used to be colleagues with. I'm still friends with someone who was a regional eBird observation validator. And this guy was really obsessed. I mean, I like birds. I take cameras and I take pictures of birds, and I never really go outdoors without binoculars on if I have a chance to look at birds. But I'm not really a twitcher. That's what birders call themselves. This guy spent all of his time doing nothing but watching birds and looking through eBird. And so these are validated by real experts. And, you can see some examples of some of the pictures. So what are we going to do today? If I go back to this, go here, and if you want to kind of look at the files, let's just peek in here a second. Don't do it right now, but the two files I'm going to go through with you, I think we just have enough time to go through these quickly, and I'll talk through them as much as I can in the time we have remaining. You'll be able to find the files. You can ignore some of the files that were just experiments that I did. I was having fun creating this, but if you were to download this folder or download the project, you don't even need the project. The ones I'm going through are this biodiversity GBIF demo.r, and I'm going through a biodiversity shiny app.r. So I'm going to show you those now. I'm going to make this a little bit bigger for people to see. I know there's some new faces in the chat. Can I just do another little survey? Can you put a 1 in the chat if you know what I'm talking about when I talk about shiny in R, or a 0 if you haven't heard of that or you don't understand what it is, and I'll just briefly explain it. Let me just see. 1 if you know about shiny, 0 if you don't know about shiny. Okay, I'm very impressed. A lot of people know about shiny. That's good. We have done quite a few things in here before on shiny. I haven't seen any 0s. Is there anybody who's not heard of shiny? Don't be embarrassed to say. Maybe I'll just say real briefly that shiny is a way to start a web server. I'm going to start a local one, but it's easy to start one on the web as well, so that other people can access your graphs and create an interactive data exploration tool. I'm just going to go through this little script just to show you what's possible with just a little bit of code. I'm very old school when it comes to shiny. I did start using it when it came out, and it was experimental. This is 132 lines of code in this app, and I'm just going to walk you through a little bit of the parts before I launch it and show it to you. Maybe I'll show it to you first, show you what it does, and then we'll walk through the parts very briefly, and I'll leave it for you if you're interested to look back. I say it's old-fashioned because there are different ways to use shiny these days, and I've used it in the most old-fashioned way, where I've created a server function, and I've created a user interface function separate, as opposed to something more modern, like there's a tool now called Shiny Dashboards, which I probably should fully embrace. I do like it and everything. So if you load this up, and if you load up the R project file, when you do, if you just all you need is, I think all you need is this R script. You should, in the latest version or close to the latest version of RStudio, get a Run App button. I'm just going to make this a little bit bigger so we can see. Just maximize that. And I just want to show you what I've done here to start with. This is a map. The map is interactive. We can zoom in and out. We can move the map around. I've chosen by default to display the first 500 observations for the parameters I set, and we'll just look at that briefly in a second, for bird observation data. The default parameters are that we're looking at bird occurrence data in Shropshire County. And if you just look in here, I'll just give you a peek. I've put a few other taxa in there. We'll explore those in a second. And just one other county to start with. It's also interactive that if we zoom in, we can click and get a scientific name, or we could replace this with anything we want. No problem. See you, Claire. Including common names or other things. And if we just want to go through and look at the Shropshire Coleoptera. Again, this is just the first 500 records, I think just for 2004. And we could keep scrolling through. Let's see what amphibians we have here. Sure, great crested newt. Common frog. Anything else? Common toad. I have it set up for just one other county as well. Now, you can dynamically grab the data. I don't have any data downloaded. You can download this app and run it if you have all the dependencies installed, and it automatically downloads when you load up the app. It's only 500 records, so it's pretty fast. You can imagine if you wanted to do something more ambitious, it would be slower, in which case it would be better for you to download the data. I'm just going to close the dash here, and I'm just going to look through just how easy it is to make an interactive app like this and grab that valuable, interesting GBIF data. So we only need to do what I did. We need Shiny for the interface. We need our GBIF for the data. I'm just going to make it a bit bigger in case people want to see it. And I chose to use Leaflet. I'm super opinionated about so many things, and I'm a bit chagrined in myself being a middle-aged guy like I am. I should probably not be so set in my ways, but I've really stopped using professional so-called GIS applications, and I much more favor 100% programmatic ones. I did start using ArcGIS before there was a graphical user interface many years ago. And I don't think it's improved now. It's really hard to reproduce things. I like Leaflet in R just for the ease and flexibility of it. And the maps are, I mean, to make a web-based map, it's far superior to other so-called professional and much easier. Not just superior in terms of efficiency, but much easier. Even beginners can make fantastic looking maps with Leaflet. Anyway, I recommend it. I've got all the stuff in the script, so it's all organized. I've got a GBIF search function here, which that I set up that creates a function called search occurrences, and I've just hard coded the country or the codes for Shropshire and Cheshire. Of course, you could add as many as you want, or other geographic centralities that are contained in the GBIF database. I've defined clade names for the taxonomy. Oops. Here, and I've limited the number of hits that I'm getting. I've limited them to a couple of the taxa, and I've coded them out to the taxon keys field in GBIF. Now, you have to know a little bit about the Darwin core data structure to take advantage of this. But the last little bit is just to do the search. You don't even need a username and password to get 500 results, and that's the upper limit. And this whole function, that's the entire definition of this whole function that creates those menus and defines those parameters for taxon and county. Then I've made the user interface with the normal fluid page, just bog standard default. And I'm not going to go through every line here because I want to go through the other script, which I think is important to demonstrate. But I've set up the inputs based on the parameters that I defined in the function just above. And that basically defines that, and the function ends there. And then we have to have another function to define the server, which I've done here. And in the server function, one of the things I'll point out is that I've coded the taxa by color, just arbitrarily. And, you could do all sorts of fancy stuff. I've made this as simple as possible just to get the idea out there. But believe me, if you invest some time in making a Shiny app with GBIF data, we are just barely scratching the surface, and there are spectacular possibilities. So you could put a key on your map, an interactive key on your map. You could do all sorts of things, but I haven't done that here. I've created a map that just uses the default background map, but of course you can change that to anything you want. I've added circle markers. That's a leaflet function to make those blue dots. You can make the colors anything you want and different colors. See you later, Joe. And really that's it. And then the last line here just launches that. So you've already seen what that does. And the last thing that I want to do in the remaining few minutes is I want to show you just a demo of getting some data at the script level. Now you can always go through the web interface. Downside of the web interface is that it's almost impossible to replicate. And the next time that you need to update your data set, if that's something you need to do, you have to go through all those steps, and it's a right pain in the bottom to get everything exactly the same so that you have a comparable updated data set. Whereas if you use our code, you literally, if you just want the new data that have come on since the last time you ran a query, you literally do not change a single character. You just run your query again and you've got all the data. So the point of this is to show you a minimum code. I've got lots of examples in here. This is also, look at that, 132 and 130. I've got lots of examples in here, but I've spent a lot of time in the data grab script here to give some what I think would be useful examples if people are interested in using this. Here, the only one we really need is our GBIF. I've loaded up ggplot. Yes, I don't even really need it, but I think I make one plot with it. And I also loaded up one called Use This. This is kind of a development package that I used, and I'll explain why when I get to that point in the script. So does anybody know what troglodytes is? Just without Googling, put it in the chat if you know what species this is. But I just want to give.
It's a wren.
It's a wren. Yeah, it is a wren. It's my favorite bird to see in my garden. So the simplest search here is we just search for the scientific name with the scientific name argument using a function, and this function is very important to keep in mind. The OCC_search is a function in GBIF that allows you to do an ad hoc search for up to 500 records with no credentials whatsoever. So it's thinking, it's trying to get some data, and boom, we've got 500. It's given me 10 lines of data. It's given me decimal latitude and longitude, and it's given me all the other, this one has 99 columns of data from the Darwin core, 500 observations, the max we can get, and we've got it all. What if I wanted to get something different from specifying just a country? Let's do that. I'm just going to demonstrate these quickly so I can get through this code in the next few minutes. So I get it again, and then we can also specify year. Notice that the year argument has got a very weird format. It's not very R-like. There are lots of weirdnesses for the arguments in OCC search and OCC download. You'll have to discover most of them yourself, and they tend to change every once in a while as well. So we get the same thing. Not that you want to do this very often, but if you're keeping track, you have a vector of observations, let's say that you made or were part of a study that you're interested in, you can grab the records by individual record identification key. There are some caveats here, and I've got a little demonstration. I'm just going to run all of this, and a plot will pop up here in a second. I'll just make the plot window bigger to prepare for it. 3, 2, 1. So we're doing a query. We're grabbing the occurrence records every year from 2000 to 2025. Oops, didn't load my ggplot function. Boy, that was a real real letdown. Let's run that again. This is one of the big deficiencies of this database. It's more important than the taxonomic bias, and it's more important than the geographic bias. And it's widely recognized and written about in the scientific literature. This is the log number of records. This is the year, every year from 2000 to 2025. And you can see there's been this exponential, literally almost a strictly linear exponential rise in the number of records. And so sampling effort is massively increased. Therefore, the number of species each year is massively increased, whether or not the actual number of species has increased or not. And guess what? It hasn't. In almost every place in this world, it's gone down. So we have to account for that in some way. We're not going to talk about that today. We're going to download some real data. This is probably the most useful thing. I'm not sure it's going to work. You have to jump through some hoops in setting up your username and password. And I came in upon some trouble when I was setting this up, and I don't have time to fix the trouble again in our remaining time. But one of the troubles that I had, I think, was I hadn't logged into GBIF for quite a while. And I don't know whether it was I needed to change my password or whether I needed to actually log into the web for this to work. But what I've done is I've created an R environ file. I'm not going to open this up because this contains the actual details of my username, password, and email for GBIF. So there are plenty of instructions online, and I'm happy to do a demonstration if people are having trouble with this. But the next thing you need to do is you need to set up your query. And then I just did a little bit of a test search, but I'm going to go ahead and do this test search. So this just asks how many wren observations are there in Great Britain for 2024, and there's quite a lot. So it says. Now I would have returned some records, and so I do have those records in there as well. I've done a test 2 that's 2023 instead, and there's a little bit less in 2023 as we expect from that pattern of bias. And then I've done a search specifying the geometry of a polygon that roughly has, this is the coordinates for, I believe that would be the north west corner of a simple box that covers Shropshire more or less. So this is, again, a very weird non-R-like format. So you can see how I did it. There are other ways to do it as well. It's all good fun. And we can see, in Shropshire, that looks too low to me, but I haven't checked it out. And then I want to try this test. This is an ambitious test. We're out of time, and I know people will have to leave. But, I think I'm actually going to call it there. I'll let people do it because I don't like going over and I know people have to leave. But what this last one does, I'll just say verbally, is that it will grab from your user environment your details. The reason I'm not going to run it is it takes about 5 or 6 minutes to run. Maybe I'll start running it and show you what I want to show you. While it's accessing, I've done this OCC download wait. Look at that. It went ahead and downloaded them anyway. Look at that. Went ahead and downloaded them anyway. Well, I'm pretty impressed. You have a zip file, you can download it. And one of the things I really wanted to mention is that with a search like this that's programmatic, and also through the web interface, you get a digital object identifier that documents your search. Of course, it's much easier to replicate with code than it is with the web interface. Just saying, again, but you do get DOIs with both of them. That's it. We're over time. I'm out of time. Thank you for coming. Hope you enjoyed this. And do, if you're interested in studying biodiversity stuff and doing more of this, because we have literally just barely scratched what is possible with GBIF and of course, visualizing biodiversity data. So if you are interested, in some way communicate that to me, and I definitely will put some more sessions on because this is a special topic of interest for me personally. I have a lot of experience working with GBIF and biodiversity data in general, and see everybody next time. Thank you for coming.